This is Sebastian Tomek. Today we will be talking about what ancient DNA is. Most of the information is from the excellent site Science Learning Hub New Zealand and the article The Half-Life of DNA in Bone, published in Proceedings of Royal Society B. Okay, so what is ancient DNA? It is a DNA extracted from old specimens, being hundreds to thousands of years old. Ancient DNA gives us a snapshot of the molecular makeup of individuals in a specimen at a certain point of time. Ancient DNA can be extracted from a wide range of source material, for humans typically preserved bones. There are several problems with this DNA due to its age. Firstly, it is often contaminated. That is, besides human DNA, you may have bacteria, etc. mixed in your sample. Scientists try to mitigate this by using a clean lab where no other DNA is handled and wearing protective clothing. Adding negative controls to monitor for introduction of contaminated DNA, for example including PCR banks. And reproducing the results, for example by using the same PCR primer on different DNA extracts from the same source material. Secondly, the quality is low due to degradation. Ancient DNA is of low average molecular size and is often damaged over time by exposure to air. This makes cloning ancient DNA difficult. Most molecular analysis uses PCR, polymerase chain reaction, to amplify the number of copies of the DNA. Once you have your sequenced DNA barcode, it is compared with barcode from known species. This is done by entering the sequence into databases like GenBank, MBL, etc. What are the limits of DNA preservation? The article The Half-Life of DNA in Bone examines this. DNA has limited chemical stability and decays without the enzymatic repair mechanisms of living cells. Following cell death, nucleases starts to cleave the DNA into fragments, and during decomposition, the DNA is digested by microorganisms. In determining long-term DNA decay, it is believed that hydrolysis of amino groups accelerates the loss of purine residues, depurinization, resulting in strand cleavage. This random DNA fragmentation generates a characteristic negative exponential correlation between DNA fragment length and number of molecules. This study fits a model for decay to 158 radiocarbon dated bones, looking on the decay of the mitochondrial DNA. An earlier claim of having sequenced DNA from an 80 to 85 million years old Cretaceous bone they find highly unlikely. The table shows the half-life depending on DNA segment length under various temperature conditions. As you can see, there is no way after millions of years any DNA would remain. At 25 degrees, DNA degrades fast, and after only a couple of hundred years there is nothing to sequence. At lower temperatures, 5 to -5 degrees Celsius, we are better off sequencing specimens tens of thousands years old. The ancient DNA samples from articles reviewed on this channel, we will see that almost all really old samples are from the North or Arctic, and very little from the tropics. And this has to do with the degradation in warmer climate. There is simply nothing to sequence. That is all for today. Till next time, I wish you all the best.